So it's my distinct pleasure to invite to the stage our President and CEO, Jonathan Simons, who we've I like to say I've now spent more time with you than Bill Nelson and Charles Sawyers did, so I'm honored to do that, and we look forward to your talk. Thank you, Jonathan.
Thank you, everyone. Thank you, Howard, for that compliment. It's a privilege to be up here, and I hope by the end of my talk, you'll get a sense of how privileged I feel and how historic I think this is for the future of the care of every prostate cancer patient and actually for the future of cancer research. And I'm going to quote the late, great first volunteer chief science officer of this foundation, Don Coffey. He would say, buckle up, because we're running a little late and we're going to drive these slides in seventh gear. I'm going to talk basically about precision with accuracy, and I'm going to do that by actually giving a perspective on 6 questions. Who's here? What's happened in 25 years? What happened scientifically in 2018 that changes everything to help for the care of patients? What challenges face PCF and you in 2019 as a part of the PCF family? What are PCF's plans to accelerate putting this foundation out of business with research, and what can you do for PCF? Who's here? Well, Michael Milken, Chris Evanson, Dr. Stuart Holden, David Eder, Ralph Feinerman, Rosie Greer, Shmuel Maytar, Henry Nordhoff, Paul Volante, Andre von Eschenbach, Neelan Youngblood, Jeff Zisk from the board. Would you all stand up? And Andrea Miyahira and Howard Sewell, would you stand up too? And it's because of them we're here. I want to thank also our many donors and friends. In the interest of time, I'm sending the slides out to 4,119 of you momentarily, but I want to thank each and every one of you, particularly our new partners and friends and leaders in the Veterans Administration. Also, just a biopsy of our board of directors, 28 of them, but 21 serve on the science committee, which we call the Discovery and Translation Committee. The medical director is the only one we've ever had, Dr. Stuart Holden. Chris Evanson has been the active chair of the science committee since the day I took the job as CEO and president. And it's really this committee meeting monthly. 75% effort of the board is supporting your research. Also, who's here are a whole set of records, so let me go. We've got 417 from universities, 116 from biopharma, 15 countries represented, 183 academic research centers, 46 biopharma companies and partners, 1 2018 Nobel Prize winner in medicine, and 1 founder and active chairman, which means a record year, a record year, a record for countries at 15, a record year for 183 academic research centers, a record for the number of biopharma partners, and, of course, a record center and record breaker in Mike Milken, our chairman, our only chairman in the 25 years of this foundation, who will make some additional remarks, which I know you're all looking forward to. In addition to that, we've been talking about N=1 exceptional responders. Here's one, $100,000 competitive award for somebody not listed as working in prostate cancer in 1997. Generated a set of CTLA-4 experimentation. This is a paper whose title I particularly love because of the micrometastatic or residual disease multimodality demonstration with CTLA-4. You'll hear from Jim later. But fundamentally, this is actually the way we roll at the Prostate Cancer Foundation. If you've got an idea as a basic scientist, translational scientist, or a clinical scientist that is of such an originality and testability, we're interested in your working in our field rather than just being in our field at the start of the year. Who else is here? Well, 11 years, 69 awards, $53 million of team science research support from the Movember Foundation. I'd give them a round of applause right now, please. And of course, we have 255 now PCF young and leading investigators. I'll submit some evidence for that leadership in a second. It's a global professional family. Of course, we ran out of land mass for the headshots 2 years ago. It's our most successful program by so many different forms of measurement, and we have never seen the field looking brighter to solving the problems for patients faster than with this cohort of human capital and true young leaders. We have 519 funded scientists right now in the research enterprise, Q4, 234 active research awards at 119 million at work, 87 teams of greater than 5 to 30 investigators in more than 3 places, and actually 63 research centers in 6 countries at the time this PowerPoint was done, which was about an hour ago. But who also is here is your unpublished data. We ran, in the last 300 days, which is the total days in the year, 82 what we call unpublished knowledge exchanges, invented actually by Michael Milken and Skip Holden at the dawn of this foundation. Andrea Miyahira, who's done such a wonderful job of this program, has had 82 unpublished data sessions during these 300 days. That's actually unpublished data of an hour or more from researchers every 3.66 days. There are a lot of things that make us special, but there's nothing more special than our commitment to getting to patients science faster by data sharing, open source but confidential sharing of unpublished data. What's happened in 25 years? I'll just give you 3 things. One is we have had a 3-log growth in the number of email addresses of this foundation. You could fit the entire research community in a large Senate dining room when Mike founded the foundation, but we got 4,119 email addresses from the last scientific retreat, and we don't have the optical capability to even give you facial recognition in the last 25% of the research community that comes to the scientific retreat. What's also happened, if you looked at our peer-reviewed discovery output table for the New England Journal, Nature, Cell, and Science, actually any 6th grader can do the arithmetic. We've had infinite growth because we had 0 number of papers in 1993 in New England Journal, 0 in Nature, 0 in Cell, 0 in Science. And as you can see in 2018, the research community has just been pounding it out for top-tier journals. Of course, this isn't adequate because we still have patients, as Maha described, every day facing the tragedy of a new diagnosis of advanced disease. But it is interesting in 2018 that June and July 2018 was like a PCF mini-series of patient bedside to BAM files and nucleotide sequence. It was as if fundamentally we were basically, we'd done a merger and acquisition of Cell and we're just pumping out your research. It was a remarkable year for basic science, but most importantly, in 25 years, top line is lives saved from prostate cancer. We, in the United States, have reduced the death rate from this disease by 52%, according to the American Cancer Society. And of course, that's backward-looking data. And we have an enormous amount of work yet to be done to put this foundation out of business. What happened scientifically in 2018 that I think changes everything for patients, everything for the basic science of this? Well, I emailed about 5 minutes ago 4,119 emails with the PROvocative 25. What I think Andrea Miyahira thinks, Howard Sewell thinks are the top 25 published papers that came in through our knowledge exchanges, and an additional 30 exceptionally awesome runners-up as well. That's Thomas Johnson hitting send in real time, so you can check your email boxes later. A few highlights. The Hanrahan and Weinberg wheel had basic science checks in all these areas. There were 8,337 peer-reviewed papers which have prostate cancer in the title. But there isn't a hallmark. I want to give a shout out to our Challenge Award and young investigators at Westwood at UCLA. This foundation creates new fields. And Owen Whitty and the discovery of reprogramming of basically normal human epithelial tissues to a common lethal neuroendocrine cancer lineage, lung epithelium, prostate epithelium, a convergence basically around reprogramming of essential transcription. We think this actually is a PCF contribution to the hallmarks of cancer potentially. I want to congratulate the basic science community, and Owen is one of the original 25-year ribbon wearers today. The PARCB, basically reprogramming, is an incredibly important insight, we think, and it's just another example of how prostate cancer can lead lung cancer research, breast cancer research, which wasn't also happening in 1993 when the NIH director said it was a wasteland for basic science and inflamed, as Dr. James Allison knows, one Donald S. Coffey. I won't cover all these, really what I think are just top-line amazing contributions to basic science, except for the following headline. Prostate cancer is now a disease of Tandem and duplications, enhancer mutations, dark DNA changes, and the paper by Quigley and others as a seminal contribution to helping us look beyond the exome. Larry Stupsky, the late Larry Stupsky's young investigator David Takeda helped us understand AR amplification by somatic enhancer mutations. It's a seminal contribution which changes permanently how we look at these patients in non-coding driver mutations. It's a bit like Orville and Wilbur, but Schultz and Van Allen submitted and ultimately published the long tail paper, which is a seminal contribution. And importantly for the citizen scientists and patients, that paper proves that if you study prostate cancer genomic drivers and start to make medicines for them, those drivers are shared by 67 other forms of chemotherapy-resistant, radiation-resistant metastatic cancer. Again, we're prostate cancer basically centric, but in the end of the day, we're cancer research leaders. And I want to just shout out one more sociologic observation. Larger than the cast of the Avengers Infinity War Part 1, which had 43 basically stars, Quigley with Feng had 16 PCF young investigators, the biggest data of the genomics of the whole genome ever done, simply as a part of our culture of sharing on behalf of patients to accelerate science. For Felix Feng, really, he could have a career in Hollywood, but we're so glad that he's the young and vigorous translational research director at UCSF. Also, that last slide, I'll go back one, that's what Don would do. If you just look at the number of young investigators as being central, not just to committing data, but actually committing interpretation, this is why team science for this foundation has become transformational in terms of changing rapidly the standard of care for patients. PCF is quite convinced that in addition to structural variations, of course, this is now a disease of chromothripsis, and a great basic science question for the PhDs is a fundamental biochemical understanding and ultimately then interception pharmacology around how you would suppress these genome structural hotspot instabilities. These are going to be as essential in preventing carcinogenesis of prostate cancer as nucleotide changes, we think, in exons, and are at the core, fundamentally, of the biology of this disease, something we couldn't have even dreamt of understanding even 5 years ago. There's no quiz for this long list of amazing 11 papers in translational science. I want to feature the Stuart Rahr Center for Precision Oncology Excellence and an Arbor contribution because it's near term for patients. I want to feature from Australia targeted radiopharmaceuticals with the philanthropic support of Stein Erik Hagen from Norway on our board at the Prostate Cancer Foundation. And I want to feature IL-23, but you have this in your inbox. All of these are very important translational contributions. As you might have heard this morning, if you're CDK12 minus somatically, you have extensive tandem duplications in your DNA. There are a heck of a lot more gene fusions with neoantigens. They're hot zone. I can't really run they're making tumors go cold to hot for T cells. These are hot tumors as a consequence of a 7% genomic alteration. And as we discussed this morning, 7% of patients we go back and see with advanced prostate cancer that's hormone refractory are potentially outstanding checkpoint inhibitor candidates. There are over 1,000 of them in the VA right now, and there are over 8,000 between North America, Europe, and Australia. And this is not a question of science. This is a question of ascertainment all of a sudden. This contribution wouldn't have been made without philanthropic support that's been led by Arul Chennaiyan, A.J. Alva now, that a remarkable group in Ann Arbor, and I think we all deserve to give them a round of applause for the 25 years, basically, of really driving a relentless interest in pathology to understand down to the nucleotide how our disease works. They're not the only ones, but they're an amazing model. Secondly, I just wanted to share with you that you can get data from the Sydney Morning Herald website of exceptional responses to a breakout new therapeutics, which at PCF we're calling quantitative theranostics as of yesterday. These are targeted radiopharmaceuticals where we're not doing case studies anymore, but we're studying dose, schedule, tumor microenvironment factors, surface expression. But this is a whole new area of therapeutics, which actually Don Coffey talked about in 1993 and 1994, and we've seen the full fruit, basically fruiting on the branches, so to speak. The PSAs are left to right. You've got 3 log changes in some patients, partial responses in other. But the point is, this field is just as real now as immunotherapy is real in our field. It's just another example of how PCF can basically invest in innovation and inspire and incent the rigorous treatment science to bring forward new treatments. It also is a demonstration of the power of social media, because all throughout Australia, these images are being shared for patients to go on clinical trials, and this literally accelerated participation in clinical trials, a case study. But the shout out here is there's a whole new challenge award category for anyone in the world working on targeted radiopharmaceuticals that are interested in quantitative theranostics. And lastly, from Great Britain and from Switzerland, for years we've talked about the tumor microenvironment as an essential part of tumor biology, but it's actionable. And really an extraordinary set of observations by Alamonte and De Bono and the group. IL-23 receptor is a target for therapeutics, but the target's on myeloid-derived cells that are CD11 positive. And what's so exciting about this is there's a deep basis in dermatology for the action of IL-23. This is another example of why PCF research is so interesting. If you told me last year I'd be talking about psoriasis and over 1,200 citations on IL-21 receptor signal activation, I'd say, really? But I wouldn't be floored. But in fact, this is very exciting, and it does bring up one other very provocative point, which is we've known for an enormous, I think a long time, that even Huggins, basically the colonization of the bone marrow, these beautiful studies by Teichmann and Pienta, it may be that what's really wired for prostate cancer bone metastases, maybe part of this is that they are enough myeloid in terms of growth factor signaling for survival that Johan and the team have really identified something very core to the core vulnerability to a fundamental property of hormone-resistant prostate cancer, which is its propensity early to colonize hematopoietic bone marrow niche. And perhaps that really is basically a JAK2 STAT3 problem. And those are very actionable targets, actually, for diseases that we haven't talked much about, like polycythemia vera. So this is a whole new dawn, in a way, thanks to this kind of team science of thinking about therapeutics we couldn't have thought of a year ago. Maha did such a beautiful job. There are so many provocative clinical contributions. I just put in orange, you should have gone to the poster session. But I do want to feature some paradigm changes. And the most important is that in a call to action, which was the strategic plan that Mike put out for this foundation 25 years ago, a goal was to get FDA-approved drugs before you had to have a metastasis, and we had to count the number of weeks you lived to get the drug approved. And this year we had not one, but two basically mission accomplished in the New England Journal, which was you could approve a drug where the target's understood, the toxicity's understood, just by measuring how much longer, if at all, it took for a metastasis to form when you knew a patient had micrometastatic disease, but you couldn't see it. I would just want to give a shout out to all those in the clinical trial consortium and in the clinical research community throughout the world. This is tens of thousands of prostate cancer patients where we never paid a penny for the later clinical development. We paid very significantly into the intellectual development and actually drug discovery and early drug development of these kinds of medicines. And what this means for precision immunotherapy, what this means for nuke targeted therapy, for theranostic quantitative radiopharmaceuticals that are targeted is we can treat patients earlier and earlier with less and less prostate cancer. It goes without saying that in the population sciences part of the PCF research enterprise, smoking is still a bad idea. Beautiful studies from the Bloomberg School of Public Health. You shouldn't smoke them if you've got them. If you have metastatic prostate cancer, this will accelerate your demise. And we're just as interested in these very significant contributions in large populations of prostate cancer as we are in somatic mutations and enhancers if they could make a difference in the life of a patient. So henceforth, we're going to be the strongest foundation, we think, other than the American Heart Association, American Lung Association, the American Cancer Society, in really educating patients that if they're smoking, they better stop. If they're diagnosed with prostate cancer, they shouldn't start. And whether or not smoking and the carcinogens of smoking really also tell us a story about the DNA damage repair defects that these advanced clones have, like they're particularly error-prone, is a provocative question that actually comes from a set of population scientists. What challenges face us in 2019? Well, I want to reflect on three. And I want to slow down for a second and just remind people of two definitions. We have a challenge in this foundation. We have actually precision with not enough accuracy. Now, statisticians know, and most second-year medical students know at least, that accuracy is the proximity of the measurement of results to the true value. And for PCF, the true value of accuracy would be 100% cure at any stage of disease. That would be absolute 100% accuracy. Precision is the reproducibility or the consistency which you get a test back that's the same as the test you did before. And at UCSF and at the University of Michigan, we can get down to the nucleotide and a billion base pairs and agree. This is amazing in terms of precision now and understanding what that could do to a gene. But if you notice, in 1993, we were imprecise, inaccurate, and we weren't in the New England Journal changing the standard of care. By 2008, we're not accurate enough in being able to give every patient, given tumor heterogeneity, and even all we know, the right medicine basically at the right time, in the right way, so we cure everyone. So if the goal of the foundation is to put itself out of business by ending death and basically suffering, we should be all about accuracy with precision. And we requires precision. I'll just go back one more time. You have to have basically precision to be perfectly accurate. So accuracy is the right treatment, the right patient, the right time. And as Dr. Hussain was alluding, we need to be right 100% of the time. And you'll notice all of a sudden conceptually that right may mean getting checkpoint inhibitors. Right may mean taking pills if you're the purple patient where you've been identified as having a tumor susceptible. Right may be getting CAR T cells if you have PSMA overexpression, or theranostics. But this right 100% of the time has an enormous number of implications. To be right 100% of the time for this foundation, or accuracy is the right treatment, the right patient, the right time, and right 100% of the time, we need the right medicine for the right target 100% of the time. And I want to just show you what keeps me thinking all the time, since I emailed it to you or at least those of you in the 4,119. We now have a PCF Precision Oncology Research and Development prescription pad. We've got 38 targets. Actually, there's a 39th, which is you can insert it and email it back to me and we'll inspect it, our team, with an unpublished knowledge exchange. And you can see that these targets are shared actually by over 60 different other forms of human cancer. And I won't give my seminar on targeting the Wnt gene today, which is one of my favorite stump speeches, for curing metastatic colon cancer through funding research at the Prostate Cancer Foundation for the 18% of prostate cancer patients that are Wnt-driven, for which Scott Takao and I do not have a medicine today. But if only 22 of 38 of these targets even have a drug at all right now, even a research drug, the implications of that are the following on the prescription pad, that 42% of what we're doing is completely inaccurate because we don't have drugs. And so if 42% of the targets are undrugged for precision accuracy with a phase 2 question to know how well that particular drug will work in that patient, what we really have fundamentally is a challenge, of drugging the currently undruggable, which is where I was last year, even though if you notice the precision prescription pad, we've added a couple pluses where we now have early drugs. So a lot of the challenge for the future of this foundation is just drugging the currently undruggable, or making our early precision drugs far more accurate. Second challenge is economic, and those are kinds of problems that it's wonderful having Dr. Michael Milken as the chairman and founder. But at our silver anniversary, PCF has teams of exceptional levels of first-in-field innovation in treatment science. Basic research all the way to the bedside, or the bedside like that miniseries in Cell back, basically bedside all the way to the bench. We've got, I think now, the largest community of gifted and talented young people from over 27 different disciplines, including bioinformaticians that take us a while to even understand. We see the output function is probably not linear, but actually now getting, it looks more parabolic in terms of PCF-funded papers getting cited every year, and that's cited by the entire cancer community. But our challenge is, with all this human capital that we've invested, 770 million raised, is that we're beginning actually at this rate to outstrip our ability to fund every single first-in-field challenge award team without more leverage on a dollar for us, because these teams are actually needing more, without more crowdfunding, which we're still experimenting with through Many versus Cancer, and partners, just people that see us as a way forward for all of cancer research. And as Don Coffey loved to say, the only thing worse than failure is uncontrolled success, which periodically was a very wise advice to junior faculty. So our third challenge, other than that of resources to genius around the world, is adding a D squared to the original CapCure 3Ds. The original CapCure 3Ds were discovery, development, delivery. The former NCI director and FDA director, Annie von Eschenbach, used these 3Ds. I think we probably got them from him. But this, basically in 1993, was we would invest in discovery, we would invest in development, we would invest in actually clinical trials to deliver so we could prove that prostate cancer could be cured, deaths could be reduced, and that was the business plan, which you could get a free book on. Skip's nodding. And thousands of these books were distributed to the corridors of power. But in 2018, we have a moral obligation to look at digital democratization of all we've learned. We ran the experiment this morning in the N=1 precision oncology session, which was so ably run by Misha Beltran. Now we're investing in discovery. We're investing in development up to the point that biopharma, we've got more biopharma than we've ever had. We're not basically chopped liver in terms of a good idea coming out of basically a PCF Challenge Award in terms of its ability to get support from venture capital or basically large corporations. But where we're deficient is paying attention to Martin Luther King's admonition that life's most persistent and urgent question is, what are you doing for others? At this meeting, I'm telling you, 7% of men, we think, with really good numbers, have CDK12 advanced prostate cancer, for which we have an FDA-approved drug, which is a breakthrough drug for melanoma and other cancers. And we do not have a digital democratization system to deliver it. And in the same way this foundation has always tried to change the course of history, we're visualizing that Siri will ask you, with digital democratization, probably with a Veterans app first, did my CDK12 gene status get checked after you checked on the weather? And how to basically democratize to patients so they can bring to the clinic the absolute state of precision and accurate genomic understanding of their cancer, we're willing to accept, but not trying to boil the ocean, but actually pilot in the VA through our Veterans Administration PCF Precision Oncology Centers of Excellence program. We don't know how to do that either, but in 1993, no one was afraid of the fact there was not basically a phase 2 cooperative group for the development of drugs. This is not a foundation that's ever shirked from an interesting challenge, and that's why we call them Challenge Awards. So, what are PCF's plans to accelerate in 2019 and put ourselves out of business? Very briefly, we're going to partner more, because basically PCF does stand for Partnerships Can Flourish. We set up an amazing partnership with an NCI very carefully over the last 18 months to study in African American men the genomic and social stressors of the disproportionate burden of prostate cancer in African American men. We provide the human capital expertise. Chris Heyman and that amazing team, including several PCF young investigators, created the grant. But in fact, we're using our taxpayers' dollars. We've committed $1 million of sort of special pilot opportunity. And for that, Ned Sharpless and the NCI have put in 26.5. And we will answer one of the grand questions which we asked in 1993 and had no answer for, is why the disproportionate burden in African American men. Second, as I've said, we're standing up. Those Captain America shields are actually centers of excellence, delivering precision oncology care. We've stood up 7 of these. We have plans for far more. Right now in the VA this year, we awarded 12 PCF Veterans Administration young investigators in the class of 2018. They're all PCF young investigators. They're all top 10% in the world, or at least in the United States, that can work in the VA. But they're all committed to getting their science in and out of prostate cancer patients, including veterans. And we will be reporting on this, although you heard several talks today from Bruce Montgomery and Matt Reddick about the dawn of this. We're creating a different kind of partnership. If we had 0.5% of ipilimumab and 0.5% of the sales of enzalutamide, we could fund 30 Challenge Awards and not even blink this year. But that wasn't what we were doing for the first 25 years. But we're taking a Ben Franklin-ology approach. We formed a partnership with Fulcrum 2020, which is a biotechnology investment company. And we would get a finder's percentage for early scouting of intellectual property out of a Challenge Award if, one, you were okay with it in the research community. We would put 100% of the ultimate return on any of that only into Challenge Award, basically back into research. The Challenge Awards will still be reviewed by NIH rules, utterly independent of Fulcrum. But it's a novel idea for inventorship, an early identification of inventorship. And Howard Sewell is at point to inform you. But do stay in touch if you just invented something amazing, of which I noted 7 things in the poster session last night. What can you do for PCF, since we're trying to do all this for patients and for the research community? Well, you can remember that 48% of the work remains ahead of us. Like Jim Allison, Charles Huggins was always interesting to listen to. I met him at 90. He was absolutely articulate that studying the immune system was a really good idea for an assistant professor trying to work on prostate cancer in 1993. And Huggins said, our work is not finished until cancer is cured, and that's basically what you can do for PCF. You can do the following. You can vote. You can participate in November 2018. And I mean participate in November 2018. You can recruit for PCF-YIs. You can collaborate in new ways, form new teams, and apply for Challenge Awards. Please inspect the PCF research and development precision prescription pad that's in your inboxes and mark it up. Mark up slot 39 and let us know what's not published, which you think is actionable or could be actionable therapeutically. There's this a whole area for how to make cold go to hot, which is probably its own rather interesting spreadsheet. You can share unpublished data and certainly RFA ideas when you do so through our UKXs or unpublished knowledge exchanges when you hear from Andrea Miyahira. Please apply in 2019 for the new NIH, DoD, and VA funds. We had a $10 million increase in the DoD allocation. That had a lot to do with congressional education out of Santa Monica. Invent and tell us early what you've invented. Engage where you can with our new precision oncology veterans programs through Rebecca Levine, whose email is provided. Join, if you haven't, Many vs. Cancer, and encourage colleagues and patients and family to celebrate birthdays by actually giving to us in honor of the birthday. And of course, say yes when we want to feature your research on PCF Digital, on PCF.org, and with donors. And lastly, and really from the heart, from all of us at the Prostate Cancer Foundation, celebrate. Raise a glass of water or any aqueous-based fluid to the silver anniversary of your foundation, pledged to go out of business with your life-saving discovery work ahead. God bless you all.